Hello, I'm here to present the effects of expanded polyQ repeats on locomotive ability in a fly model of Huntington's disease. So to start off, Huntington's disease is a hereditary neurodegenerative disorder caused by mutations in the Huntington gene that result in expanded polyQ repeats. The length of the polyQ repeat dictates the age of onset and severity of disease. So individuals expressing 36 polyQ repeats or less will not develop HD, whereas expression of more than 40 repeats will develop HD pathology. The more repeats you develop, the more severe the disease is and the earlier the age of onset is. The polyQ repeat causes aggregation of the Huntington protein, which causes progressive neuronal death. Symptoms associated with HD include uncontrollable motor movements, dementia, and mood swings, and the disease is lethal in all cases. So animal models such as Drosophila are commonly used to study neurodegenerative disorders such as HD. The UAS-Gal4 driver system allows you to selectively express a gene, and to selectively express in neurons, we use the ELAV driver specifically. The way that locomotion is studied in flies can be done using behavioral assays such as the negative geotaxis assay. This assay utilizes the fly's natural tendency to climb upward in response to a startle. So in the current study, the locomotive ability of Drosophila expressing either 16 or 128 polyQ repeats was tested using the negative geotaxis assay to determine if the expanded polyQ repeat has an effect on motor behavior. We hypothesized the expanded polyQ repeat of 128 glutamine residues would increase the time required to complete the negative geotaxis assay. To study the effects of expanded polyQ repeats on fly locomotive behavior, we used an ELAV-Gal4 UAS polyQ system to express polyQ repeats of either 16 or 128 glutamine residues in the flies. So for this system, we took ELAV-Gal4Y males and crossed them with UASQ16 CYO females or UASQ128 UASQ128 females. The progeny expressing either polyQ16 and polyQ128 were aged for 2 weeks, and then we tested them using the negative geotaxis assay. So for this assay, we had 4 flies from the polyQ16 group that functioned as our control because they do not develop the symptoms of Huntington's, and then we used 4 flies from the Q128 group as our treatment. 4 flies were tested per group, and 1 vial was tested at a time. The time it took for at least half of the flies per group to reach the top of the vial was recorded, and 12 trials were taken. The time was capped at 20 seconds. After we took our data, we calculated the means and standard deviations for each group. On average, the flies in the Q128 group were found to take longer to complete the negative geotaxis assay compared to the Q16 group. The mean time for the Q16 group was 8.33 seconds with a standard deviation of 2.37 seconds, while the mean time for the Q128 group was 11.42 seconds with a standard deviation of 2.61. The results show a positive correlation between the polyQ expansion length and time to complete the negative geotaxis assay. However, no statistical significance tests were performed to analyze these data, so we can only say that there's a correlation between the 2 variables. The aim of the current study was to evaluate the effects of expanded polyQ repeats on motor function in a fly model of Huntington's disease. So on average, the expanded polyQ repeats of 128 glutamines, which is well within the lethal range for Huntington's disease symptoms, impaired motor function. So in general, these flies showed less motor control and less efficient motor skills, which parallels the human condition. However, some limitations of the study include small sample sizes. For example, 1 fly in the Q16 group showed impaired motor behavior from the very beginning, likely due to a damaged wing during fly transfer. So our small sample size was unable to combat for these individual differences, which, if this experiment were to be repeated, a larger sample size would be ideal to control for these individual differences. The findings of the study show that expanded polyQ repeats result in motor impairment observed in HD. So further research should be conducted to investigate the mechanism of how the expanded polyQ repeat results in neuronal death and motor impairment. Current research points to aggregation of the Huntington protein as a possible mechanism underlying neuronal death in HD. So understanding the expanded polyQ repeat and how it causes aggregation of the Huntington protein at the cellular level is imperative for identifying therapeutic targets for the treatment of HD. Lastly, I just want to thank my wonderful lab partner, Hannah Lynch, for her support and partnership throughout this project, and I'd also like to thank Dr. Casey and our PIs for their continued instruction and help throughout the semester. Also, I just want to thank my TAs, Casey Lamar and Anna Henkin, for their continued instruction, guidance, and support. Thank you.